Good day, this is the Professor, drwillphd, PhD, and today is Saturday, November 26, 2022. It is 5:37 and 23 seconds, a.m. Eastern Standard Time, and today's research, artificial DNA. So let's hop to it here. Artificial intelligence tailors artificial DNA for future drug development. Date November 25th. 2022. Source: Chalmers University of Technology. With the help of artificial intelligence, researchers have succeeded in designing synthetic DNA that controls the cell's protein production. The technology can contribute to the development and production of vaccines, drugs for severe diseases, as well as alternative food proteins much faster and at significantly lower cost than today. How our genes are expressed is a process that is fundamental to the functionality of cells and all living organisms. And simply put, the genetic code in DNA is transcribed to the molecule messenger RNA, which tells the cells factory which protein to produce and in which quantities. Researchers have put a lot of effort into trying to control gene expression because it can, among other things, contribute to the development of protein-based drugs. A recent example is the messenger RNA vaccine against COVID-19, which instructed the body cells to produce the same protein found on the surface of the coronavirus. The body's immune system can then learn to form antibodies against the virus. Likewise, it is possible to teach the body's immune system to defeat cancer cells or other complex diseases if one understands the genetic code behind the production of specific proteins. Most of today's new drugs are protein-based, but the techniques for producing them are both expensive and slow because it is difficult to control how the DNA is expressed. The researchers took an important step in understanding and controlling how much of a protein is made from a certain DNA sequence. And first it was about being able to fully read the DNA's molecules instructions. Now we have succeeded in designing our own DNA that contains the exact instructions to control the quality of a specific protein. The principle behind the new method is similar to when an artificial intelligence generates faces that look like real people. By learning what a large selection of faces look like, the artificial intelligence can then create completely new but natural-looking faces. It is that easy to modify a face by, for example, saying that it should look older or have a different hairstyle. On the other hand, programming a believable face from scratch without the use of artificial intelligence would have been much more difficult and time-consuming. And similarly, the researchers' artificial intelligence has been taught the structure and regulatory code of DNA. The artificial intelligence then designs synthetic DNA where it is easy to modify its regulatory information in the desired direction of gene expression. Yet, simply put, the artificial intelligence is told how much of a gene is desired and then prints the appropriate DNA sequence. The DNA is an incredibly long and complex molecule. It is thus experimentally extremely challenging to make changes to it by iteratively reading and changing it, then reading and changing it again. And this way, it takes years of research to find something that works. Instead, it is much more effective to let an artificial intelligence learn the principles of navigating DNA. What otherwise takes years is now shortened to weeks or days. The researchers have developed their method in the yeast Saccharomyces cerevisiae, whose cells resemble mammalian cells. The next step is to use human cells. The researchers have hopes that their progress will have an impact on the development of new as well as existing drugs. The protein-based drugs for complex diseases or alternative sustainable food proteins can take many years and can be extremely expensive to develop. And some are so expensive that it is impossible to obtain a return on investment, making them economically non-viable. And with our technology, it is possible to develop and manufacture proteins much more efficiently so that they can be marketed. And this is a professor of microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.